In this video, we are going to study the structure of secondary oocyte. The secondary oocyte is also called as unfertilized ovum. It is released from the ovary during ovulation. It having a outermost covering is called as corona radiata cells. These corona radiata cells are glued with each other by hyaluronic acid, and below to this corona radiata cell there is another layer which is a very transparent non-cellular membrane called as zona pellucida. This zona pellucida, it avoids wrong site implantation. Then just below to it there is a vitelline membrane, or it is also called as the plasma membrane. In between the plasma membrane and the zona pellucida layer there is a space called as periviteline space, and this periviteline space, which is fluid filled cavity, and here you can see here is a first polar body is also present, and the plasma membrane forms a boundary around to the cytoplasm. The cytoplasm is divided into two parts, the ectoplasm and the endoplasm. The ectoplasm is granular one, which contains a cortical granules, and the inner one, it is clear one. Endoplasm is clear. Okay. The secondary oocyte in case of human or the human egg is microlecithal type. That means it having abundant amount of cytoplasm and very little amount of yolk. As it contains so much cytoplasm, this cytoplasm pushes the nucleus towards the periphery. So the nucleus of secondary oocyte is placed at periphery, and it is basically near to the polar body, at the site of polar body. The nucleus is also called as germinal vesicle. This germinal vesicle or nucleus is big in size, which contains a chromatin network. Especially, it is arrested at metaphase. It is arrested at metaphase. Very important question can be asked for NEET or CET. It is arrested at metaphase. So this is all about the structure of secondary oocyte, or it is also called as unfertilized ovum.